We've recognized that gastric cancer is a heterogeneous disease, and there are unique subsets of tumors based on histology, clinical behavior, and biology. And now the Cancer Genome Atlas published, and we've established that molecularly these subtypes are quite distinct. So that's where the field has evolved into, beyond the WHO classifications or just histologic classification, is really to recognize on the molecular level that these tumors are distinct. And so we see that esophageal squamous cell cancer and adenocarcinoma of the esophagus are molecularly different and should never be approached in the clinical trial in a same trial for the same patient population. In fact, the squamous cell of the esophagus biologically is very similar to very aggressive types of head and neck squamous cell tumors. Within adenocarcinomas, esophageal adenocarcinomas biologically are similar to G-junction tumors, and therefore these tumors can be used and put together into similar clinical trials if we're developing molecularly targeted agents. These tumors tend to be chromosomally unstable, have a lot of amplifications in the receptor tyrosine kinase, such as HER2, EGFR, or MET. They're also p53 mutants, so they tend to be more aggressive and grow and metastasize at an earlier time in the disease point. And also, these tumors tend to be non-MSI, although what's important to note is that we can't guess based on anatomy if this is going to be an MSI tumor or an EBV tumor, or and really histologic and molecular characterization is quite important. So chromosomally unstable, and then the three other subtypes, biologic subtypes, are genomically stable tumors. Those are the signet ring cell histologically most commonly, and those are the claudin rearranged tumors, row A, ecoadherent altered tumors, and we'll talk about claudin as a possibility for targeted agents in the future. And MSI and EBV subtype within the TCGA are the most provocative and important for targeting with immunotherapy. EBV positive gastric cancer is rare but very important to study and to diagnose, as 80% of those tumors are PD-L1 positive. And then within MSI, the indication for immunotherapy is quite strong, and responses can be quite dramatic. And again, although these tumors are rare in metastatic patients, they're important to look for, and as they will dramatically change the treatment algorithm.